Hello, what's going on, Grecians? Welcome back to another episode of The Scientific Mind with Sir Eman. Last episode, we discussed the topic about cardiorespiratory diseases, which is the last topic for the first chapter, all about respiratory system and circulatory system working together. In this episode, we are going to start the chapter 2 of the first quarter, which will be focusing on heredity, inheritance, and variation. To begin our discussion for today, let's have our prayer. Before we proceed our discussion for today, let me show you students the activity that you need to accomplish for this week. Okay, the first activity that you need to accomplish is the boy or girl. In this activity, you need to draw a Punnett square which shows the inheritance of the sex chromosomes. Represent the female sex chromosome with XX and the male sex chromosome with XY. After doing the Punnett square for the inheritance of the sex chromosomes, you are going to answer the 6 guide questions. So you are going to answer this on Tuesday. The second activity that you need to answer on the third day is the activity entitled When Gender Matters. So you need the activity sheet, this one, and pencil. Using a Punnett square, you are going to show the probability of the children who will have a normal vision and children who will be colorblind if a colorblind man marries a woman who is a carrier of the colorblind gene. After doing the Punnett square, you are going to answer the 5 guide question. Also, after finishing that worksheet, you are going to answer this concept map about heredity and answer part B, multiple choice, items 1 to 5, and answer the question on part C, item 1 to 3. Okay, so for the last day, you are going to answer concept mastery, multiple choice, and the identification part on page 77 to 78. And you will research about the disadvantages and advantages of inbreeding. Make sure to finish all those activities and assignments before the deadline. Okay, so without further ado, let's start this discussion. Our topic for this episode is all about sex determination and sex-linked traits. When a mother knows that she is pregnant, she will always go to the doctor for a checkup. One of her checkups includes an ultrasound where she can see the image of the fetus in her womb. In addition to that, if she is 4 to 5 months pregnant, she can determine the baby's sex. Now, how does a fetus acquire its gender? To answer that question, we will go over this sex determination topic. But before we answer that question, let's have an overview about the human chromosome. Chromosomes consist of a material called chromatin, which is composed of a mixture of DNA, or the deoxyribonucleic acid, and structural proteins called histones. The DNA in a given chromosome is one long unbroken nucleic acid molecule. Chromosomes consist largely of genes, which are lengths of DNA that carry chemical code for a particular protein product that is synthesized elsewhere in the cell. This particular protein includes adenine, guanine, thymine, and cytosine. For the most part, Genes on the same chromosome bear no relationship to one another in terms of the protein they code for. Sex chromosomes, also known as allosomes. These chromosomes contains the genes that determine the biological sex. Chromosomes that differs from one ordinary autosomes in form, size, and behavior. The human sex chromosomes is a typical pair of mammal allosomes which determine the sex of an individual created in sexual reproduction. How sex is determined? Sex determination is the process of sex differentiation by which whether a particular individual will develop into a male or female sex. Sex expression is governed by chromosomes and genes. In unisexual animals, chromosomes are of two types, allosomes and autosomes. Autosomes are chromosomes which do not differ in morphology and number in male and female. Allosomes are chromosomes which differ in morphology and number in male and female, and it contains the genes that determine the sex. Unisexual diploid individuals have two sex chromosomes, and the rest are autosomes. Mechanism of sex determination. There are four important mechanisms to determine the sex of an individual. First is the sex character, 2. Chromosomal sex determination, 3. Monogenic sex determination, and last is the environmental sex determination. The first mechanism in determining the sex of an individual is the sex character, which includes the primary and secondary character. Primary characters includes the gametes, which are the sex cells, while secondary character involves all the genitalia or the physical features that determine whether an individual is a female or a male. Next mechanism is the chromosomal sex determination. In a diploid individual, there are 2n-2 autosomes and 2 sex chromosomes. There are three types of chromosomal sex determination. First, allosomal sex determination. Second, Diploid-Haploid system of sex determination. And last, the genic balance system. Let's tackle first the allosomal sex determination. Allosomes or sex chromosomes are generally X and Y types, but in some birds, they are of Z and W types. Sex with similar types of sex chromosomes or the XX is known as homogametic sex. Homo, which means the same, gametic sex cell. And with the similar type of sex chromosome or XY is known as heterogametic. Hetero means different, gametic sex cells. There are four different systems under the allosomal sex determination: the XXXY female-male system, the XXXO female-male system, the XOXX female-male system, and the ZWZZ female-male system. Okay, so let's first discuss the first system in the allosomal sex determination, which is the XXXY female-male system, which are common in Drosophila melanogaster or the fruit flies. This system is also found in humans and some other mammals. In this system, female has two X chromosomes, which is homogametic, and produces only one type of gamete or sex cells, which is the X, while the male has one X and one Y chromosome. So it is a heterogametic and produces two types of gametes, the X and the Y. So how this system will produce or determine the sex of an individual? If an X ovum meet with X sperm, it will develop into a female sex with a gamete or chromosome of XX. If the X ovum unites with the Y sperm, it will produce a male or the XY sex. The second system under the allosomal sex determination is the XXXO female-male system, which are common or mostly found in grasshoppers and many species of Orthoptera and Hemiptera insects. When we say Orthoptera, they are the insects that have straight wings, while Hemiptera are insects that sucks blood or feed on blood. So what is XXXO female-male system? In this system, female has 2 X chromosome and male has 1 X chromosome. Female is homogametic and produces all the eggs with X chromosomes. Male is heterogametic, which produces sperms, half of which have X chromosomes and the other half have none. So how this system produce the male or the female? The union of egg with sperm having X chromosome will give rise to a female sex or the XX, while the union of egg with the sperm having none results in the development of male sex or the exogamete. Okay, now let's proceed to the second system in determining the sex under the autosomal sex determination, which is the XOXX female-male system, which are found in very few species of insects like Pomea. So Pomea are species of moth. The third system under the autosomal sex determination is the XOXX female-male system, which are found in very few species of insects like Pomea or species of moth. In this system, male has only 1 X chromosome and hence female is heterogametic. As a result of meiosis, 50% eggs of such female carry an X chromosome and the remaining 50% have none. On the other hand, male has 2 X chromosomes and produces all the sperms with 1 X chromosomes. Thus, male is homogametic. The union of X sperm with ovum having X chromosomes will give rise to male sex, while the union of X sperm with ovum having none leads to the development of female. The last system of autosomal sex determination is the ZWCZ female-male system, which are found in birds, butterflies, and moths. Female is heterogametic and produces 2 types of gametes, the Z and the W types, while male is a homogametic and produces all the sperms of the same type carrying 1 Z chromosome. The union of Z sperm with ovum having Z chromosome gives rise to male, and the union of Z sperm with ovum carrying W chromosomes leads to the development of female sex. The second types of chromosomal sex determination is the Diploid-Haploid female-male system, which are found in honeybees, ants, and termites. In honeybees, the females have diploid, or 2N, which is equals to 32 chromosomes, and drones, or the males have haploid, which have 16 chromosomes. Females are the queens and workers. The females which fed on royal jelly develops into queen. When the queen is fertile, the workers are sterile. The queen produces haploid eggs. However, haploid male bees produces haploid sperms by mitosis rather than meiosis. The union of eggs with sperm gives rise to diploid larvae, which become female. Okay, the third type of chromosomal sex determination is the genic balance system. According to this theory, sex of an individual is determined by a balance between the genes for maleness and for those femaleness present in an individual. Genic balance theory states that sex-determining genes are present on both sex chromosomes as well as on autosomes. The male sex-determining genes are present on autosomes and female-determining genes are present on X chromosomes. The sex expression is determined by the balance of genes on autosomes and X chromosomes. In other words, the expression of sex depends on the ratio of X chromosomes to that of autosomes. This ratio is represented as X over A ratio, where X means the X chromosomes and A as autosomes. Okay, the third mechanism of sex determination is the monogenic sex determination. It is the expression of sex which is influenced by a single gene. In Drosophila, a transformer gene or TRA which is present on autosomes plays an important role in sex determination. Transformer gene is recessive, hence does not have any effect in heterozygous condition on either sex. In homozygous condition, this gene transforms the normal diploid females into sterile males. The fourth and the last mechanism of sex determination is the environmental sex determination. Sex determination in some organisms such as seaworm, Bonellia and Denopulus, and horsetail plant is governed by environmental conditions and also includes some hormonal effects. In Bonellia, the larvae which remain free in the sea water and settle on the sea bottom are differentiated into females, while those larvae which settle on the proboscis of female develops into tiny males. Sex-linked traits. Sex-linked traits are controlled by the genes found in the sex chromosomes, the X chromosomes. Example for this is colored blindness and hemophilia. So what is hemophilia? Hemophilia is characterized by delayed blood clotting or coagulation due to the absence of clotting agent called factor VIII. Since males XY have only one X chromosome, they are affected by the recessive X-linked traits more often than the females or the XX. So let's try this. Using a Punnett square, illustrate how colored blindness is inherited when a carrier woman marries man with a normal vision. Let X with exponent of capital B stand for normal X chromosome, X with exponent of small b, the carrier chromosomes, and Y, capital Y for normal Y chromosome. So this is the Punnett square. Now put the allele in the top part of your Punnett square. Put the allele of the carrier woman, X capital B and X with a small b. For man with normal vision, normal X chromosome, then normal Y chromosome. So cross these two allele. Cross these two allele, which will have X capital B, X capital B. For this part XB, X small b. For this part, normal X chromosome and the normal Y chromosomes, X exponent of capital B, then Y. And for this part, X small b, Y. So let's determine the phenotypic ratio and the genotypic ratio of the offspring between. Now let's determine the phenotypic ratio of the offspring of a carrier woman that marries a man with a normal vision. Phenotype: 50% normal child. So where this 50% come from? If you will look at your Punnett square, the 50% normal child is represented by this part, X capital B, X capital B, and this part. Since each box represents 25% and there are two boxes, 25% plus 25% equals 50%. Another phenotype is 25% carrier child. In this part, X with the small letter B is the carrier. So this is the carrier, which is only one, box 25%. And the last one is the colorblind child. This one, 25%. Again, why this part is a colorblind? Because male has only X chromosomes, therefore it will be affected by the X-linked trait. Now let's determine the phenotypic ratio of the offspring on the basis of gender. Let's tackle first the male. Phenotype, 50% normal and 50% colorblind. Why 50%? So we are going to look only at this part, the part with have X and the Y chromosomes. Since it has only two boxes, each box will represent 50%. Therefore, this part which have capital B, which is normal, therefore it will have a normal 50%. It is 50% normal vision, while this part is a color blind because of the B, that small b that represents the X-linked traits. So 50% colorblind. Now for the female, we are going to look at only this part, the part with the XX chromosomes. Again, this part is only divided into two parts, therefore each box represents 50%. The phenotype is 50% normal, represented by X with a capital B as exponent. This is the normal vision. Then this part with a small v will be the carrier. Why it is not a colorblind? Because the X-linked traits in female is recessive when there is only one. So these X-linked traits will only become a carrier gene, and it have a ratio of or a value of 50%. Next is the sex-influenced traits. These traits are expressed by a particular sex. These are the genes that produce sex-limited traits. These are genes that produce sex-limited traits that are mostly found in autosomes than sex chromosomes. An example of this is bird. Genes for heavy bird is only expressed in males, but both male and female can be a carrier of this gene. In sex-influenced traits, most of the genes on the X chromosomes in humans have no counterpart on the Y chromosomes. Thus, these genes or any genes on the X chromosomes are expressed in males even they are recessive in females. Traits or the X-linked traits influenced by these genes manifest the following characteristics. First, these traits cannot be transmitted from father to son. Carrier females, or those who carry the genes on one of their X chromosomes, have a 50% chance of passing on these genes to their offspring. And the last, father will pass on the genes to their daughters, who will become the carriers of these genes. Next is the sex-limited traits. So these are genes that are dominant in one sex and recessive in another. Baldness is only dominant to men. Women can only have baldness if they have two alleles of these dominant traits. That ends our episode for today. This is the Scientific Mind with Sir Eman. I hope you learned something from today's discussion, and goodbye.